I think it's a really beautiful representation of how the porphyrin molecule, this really delicate molecule that if it's exposed to light produces really horrible reactive oxygen species that destroys things. It's very self-destructive. So this piece of work shows how the porphyrin is held in place and is carefully transported. My name is Dr. Nate Adams. I'm a research scientist and science communicator. I work in the molecular biology field, biochemistry, chemical biology, and I research the early stages of chlorophyll biosynthesis. I'm also a science communicator, so I spend a lot of time performing and communicating and working with other artists and collaborators to create different ways of talking about science. With our first collaboration with Seiko, it was very much a very literal interpretation of some scientific work. And I suppose my main role now is just to make sure it's scientifically accurate. Seiko comes with these really intricate questions whether, are there enough turns in this helix? Is this molecule precisely correct? And our collaborative process itself, well, it's mainly us drinking a lot of really strong black coffee, getting really over-caffeinated and discussing science and art.
My name is Seiko Kinoshita. I'm an artist. I've been doing about 20 years and I like making quite large installation. I started as a textile artist, but nowadays I use all sorts of different materials.
So I started working with Seiko for the 2015 Krebs Fest, which was a big celebration of biosciences in Sheffield, where we created a large-scale installation, or what we thought at the time was a large-scale installation, called the GFP, which is now housed at the University of Sheffield. And then from that, we really enjoyed working together. So we put together a grant application in 2016 for Festival of the Mind, which saw us build a huge large-scale origami, steel and wood sculpture called the METABOLON, which we exhibited in the Sheffield Cathedral. That was really good fun, and it was just a really good and exciting and amazing piece of work.
METABOLON, first time I met with Nathan, in his research, they talk about folding, protein folding a lot. That folding word stayed in my mind, and I thought, oh, maybe I can do origami relating to that, his research. And as well as that folding, those kind of repetitive process, I thought it's really showing the scientific researchers' everyday life. They repeat again, same process. Mainly, we've been working with Darren Richardson. He's great, he's an artist as well. So it's really nice working with, three people mainly, Nathan, me and Darren. We can put all our ideas together to make it something more interesting and accessible for people. For Aegis, we really wanted to make the helix structure correctly. So we thought that origami is not really a good idea. So in this time, I really wanted to use a textile technique. I dyed all color and I wove myself on the Adobe room. That Adobe room is 100 years old. Again, it's a really repetitive process makes something really one big installation. I'm really hoping people can see it's kind of like his research effort that's making to the final, his research paper. That's kind of showing in my artwork.
Doing this sort of work is important for a number of reasons. From a personal satisfaction, I really love producing different types of artwork and being creative. But also the fact that all of this work is funded by the taxpayer, and fundamental enzymology isn't the most accessible subject, but I believe that if we can produce work that communicates to an audience, connects with an audience, that tells them about what's going on in the plants or the cells or the algae around them, they can get an idea of what is occurring at the nanoscale. And while a lot of it, the maths, the science, it's irrelevant. You don't need to know what's going on in terms of, the actual hardcore science. But the story behind it, the understanding that we're slowly picking apart one of the most fundamental processes that allows life to survive on the planet, I think that's an important story to tell. And also it's quite beautiful, the structures, the function, everything working together is kind of amazing. During lockdown, due to the pandemic and the fact that lots of projects had to pivot because we didn't have the opportunity to exhibit, one of the things that we've done is actually produce a better digital archive of the work. We put Aegis up to be used as a backdrop to do a different project called Physical Education, where we've been using circus arts to explain the chlorophyll biosynthetic pathway using aerial.
I'm Claire, also known as Madam Mango on stage. I don't think I have performed to a piece of art before or with it as a backdrop. And I think for me, in this case, it was really relevant. It is what I'm talking about, which is the creation of magnesium protoporphyrin 9 using text and aerial. And here we have just a beautiful visual representation of it.
Luckily enough, I'm an aerial artist as well as being a scientist. And yeah, I got to perform with Aegis as well. I was just as a backdrop, and I'm talking about molecular motors and everything that goes on, and I just really love it. It was quite inspiring having my rope just there next to Aegis and performing. Really loved it. So I think we've been really lucky to be able to pivot the project to allow us to do this during this time.
I wanted to show the possibility of what we can do, art and science together, because each of them completely different subject. Normally we see opposite, but we can put it together, much more interesting things and exciting for the people, I hope.